Hey everyone, today's guest is Dr. Li Cheng, who is a professor in the Department of Pharmacology at Peking University in Beijing, China. Today, Dr. Cheng will present his lab's findings for the impact of immunoglobulin G on extending lifespan and on obesity. So welcome Li, and let's get into the presentation.
Okay, thank you, Mike, for the invitation. Let me share my screen. I think it's really important to have this opportunity to share scientific findings with the public. And by the way, Mike, I really like the slogan on your t-shirt, "Conquer Aging." That's our goal. Yeah, to make everyone live longer and healthier. Let's talk about aging and obesity from a perspective of IgG. I'm trying to use IgG to put these two together. I believe most of us know what is aging and obesity. And right now, this is maybe the first time in human history that people that are over 65 years old are more than children under 5 years old. That means that our human society is going to have an aging future. And also that this is an obese world. And right now there are over 1 billion people overweight or obese. And in the US, that's even worse. US, that only less than 1/3 of people have normal weight. So these two, an urgent public health crisis now. So indeed, aging and obesity are so similar to each other in terms of their comorbidities, such as cancer, cardiovascular disease, neurodegeneration, and liver disease, so on and so forth. In another way, aging and obesity overlap with each other by the hallmarks, like chronic inflammation, oxidative stress, senescence. So these two are so similar to each other. It's hard to not believe these two are not closely connected. That's our research focus on. I try to understand whether they have a common mechanism. So all the known famous longevity pathways, they are all critical for metabolic regulation. And the current anti-aging interventions all involve changing metabolism. So there's we view aging from a metabolic perspective. So our question is actually quite simple. We try to understand, why do we become obese when we age? And this question is not very common. Like for myself, I can eat whatever I want in my 20s. But after 30s, I have to be so careful to watch what I eat just to keep in shape. Also, aging and obesity develop so many same comorbidities. Do these comorbidities have the common mechanisms? Therefore, we can have a therapy for more chronic diseases. That's also our goal. So we start from here, and this is a famous experiment that initiated in 1950s, that researchers connect an aged rat with a young rat. And they found something really striking, that these aged mice, that after sharing their circulation with the young mouse, they become younger. So the people are so fascinated by something called a longevity factor in our circulation. So people have been delved in this for decades, try to identify longevity factors. Interestingly, in this experiment, on the other hand, that people also notice that this young rat actually aged faster after sharing the circulation with this aged rat. So therefore, that indicates that there is something in the circulation also drives aging. That's an aging factor also in the circulation. So whether it's a longevity factor or aging factor, that's people are keep trying to identify and answer the question. So for us, Our lab used to work on metabolism. I've been working at Columbia over 10 years, running a lab focused on adipose tissue and metabolic diseases. So we did a simple experiment, did this gene expression profiling across different tissues using mouse models. That's very common in research. That we found that so these age-related genes are accumulated over age. That's all we expect. And interestingly, that they're across different tissues, that the highest age-related genes are all in adipose tissues. This one, we call it epididymal fat, is equal to the belly fat in humans. The second highest one is subcutaneous fat. The third one is brown fat. They all accumulate the highest age-related genes, indicating that adipose tissue is an tissue that prone to aging. Something also interesting that this aging-related changes also happens at the earliest time point in adipose tissue than in any other tissues, indicating that adipose tissue is the driver of aging. We also have a study in collaboration with Dr. Lei Sun from Duke NUS at Singapore. That we introduced a dynamic way to analyze gene changes during aging. We found that the during aging, what causes the metabolic decline is not the absolute gene expression level. Instead, it's the response. So during aging, the genes involved in the metabolic regulation to stimuli is impaired. Therefore, they cause the overall decreased metabolic function. So we call this the impaired metabolic elasticity. Interestingly, in this main metabolic tissues, like in the muscle, liver, fat, adipose tissue has the worst impairment in metabolic elasticity. Therefore, we further believe to focus on adipose tissue to identify the trigger that cause adipose tissue aging. We started from some really simple experiment. We use mice. We use mice focus on their adipose tissue to compare the different change, the different protein change over at different ages. And we first did this metabolic the mass spec, try to measure all the proteomic changes. And we found that in adipose tissue over age, the highest changes actually are all the proteins are related to immunoglobulins. And these proteins are all at the highest abundance. So look at this, we call it Western blot, that is showing the protein abundance. You can appreciate that in this 8 weeks, very young age of mice, they have really low antibodies, IgG, in their fat. Well, when they are about 1 year and a half, this is like early aging time to humans, that this accumulates so much IgG. And this accumulation happens really early in life. At 26 weeks, at about half-year-old of age in mice, equal to about 30 years old in humans, that IgG start to accumulate in fat, in mouse fat. And further, please note that you may see that okay, this IgG is come from circulation, because in our blood there is so much antibodies there to protect our body from pathogens. Yes, antibodies do increase during aging, like in mice, it increases several fold, but not as high as in adipose tissue. Adipose tissue is far more sensitive in terms of IgG accumulation during aging. Also, if this IgG leaks from circulation, we may expect the same level of IgG in different tissues, but indeed it's not. Look at the IgG levels across different tissues, like in fat, liver, muscle, kidney. the brain and heart. Adipose tissue have the highest IgG, both in heavy chain and light chain, that indicating that adipose tissue have a special mechanism to enrich IgG over other tissues. You may also appreciate that in this time course in different tissues that adipose tissues start to accumulate IgG earlier than other tissues. Other tissues also have IgG accumulation in late aging, but they start at a lower at a later time point, and also at accumulate at lower levels. So adipose tissue is really the key to connect aging and metabolism. People are trying to develop some anti-aging interventions. So far, the calorie restriction is the most efficient way for aging intervention across different species. In this famous experiment that did by scientists from Wisconsin medicine researchers, that they put monkeys under calorie restriction over 20 years. And then they found really interesting. The monkeys under calorie restriction over their lifetime do look much better when they aged. Look at their fur. So they claim that calorie restriction can increase healthspan. So we also adopt a calorie restriction to see if IgG is an aging-related factor. Does it change by during calorie restriction? Then something really surprising to us happened here. We found that when we put mice under calorie restriction, that it specifically decreased IgG without affecting IgA or IgM or other antibody isotypes, meaning that IgG is specifically regulated by this by calorie restriction. So this is in the plasma. IgG was dropped, decreased over 50%, and it was even more pronounced in adipose tissue by calorie restriction. So further make us believe there is a connection of immunoglobulin and aging. So what is the IgG? This is the most abundant antibody in our body, accounting for over 80% of our total antibodies. It also have the longest half-life, over 3 weeks. And these are all the major IgG isotypes here. IgG have this as antigen recognizing domain called FAB, and also have this constant fragment called FC fragment here. So this is to recognize antigen. This is to activate its downstream receptors. So we all know that antibodies are for immune protection. So the more the better. So it's so to test that, we just as a purified antibody, total antibody from young and lean mice, healthy mice, just to inject the antibodies, IgG, to another healthy mice, just to increase the amount of antibody to mimic what happened in aging. Then we notice that after IgG, after we simply boost the IgG levels, it impairs the body's response to insulin. We call it insulin sensitive, insulin tolerance test. This is something we usually measure our body's response to insulin. So we give a bolus of insulin to humans or to mice. We measure how quick the glucose drop. So after given IgG, it delayed the drop of blood glucose. So something happened bad in the body. We also noticed that with this IgG treatment, it accumulated in adipose tissue, and also it increased a protein called F480. This protein is the marker for macrophage, which is the scavenger immune cells in our body. So IgG increase macrophage. That made us speculate that IgG may also cause inflammation in adipose tissue. So again, we did a transcriptomic analysis of adipose tissue after IgG treatment. We noticed that there are so many pathways are activated by IgG treatment. Most of them are inflammation related, inflammatory response, neutrophil. So that tell us that IgG is not just accumulate in adipose tissue, it also does something there, something bad, because pathogenic change in adipose tissue and also in the whole body metabolism. Here, I mean insulin sensitivity. So how does IgG cause this impairment of metabolic function and adipose tissue function? And we know that adipose tissue also undergo fibrosis. That means we have too much collagen deposited in the tissue. That will make the tissue rigid. Then it will worsen inflammation and also impair insulin sensitivity. We also notice that adipose tissue fibrosis also take place in aged during aging. This is the cells red staining for this collagen. It's a beautifully red color, and also under polarized light, it you can see fluorescent, very beautiful under microscope. So it's with this prevalent fibrosis in adipose tissue. However, when we checked a mouse model we call the B-now mice, these mice have a deficient B cells, therefore they cannot produce IgG. These mice are protected from adipose tissue in aging. They have much less cells red staining and polarized light imaging here. So then that tells us that IgG might be related to tissue fibrosis. So we know fibrosis is driven by a major pathway that the TGF-beta signaling that can activate the downstream SMAD2 and SMAD3. So in B cell now mice, without IgG here, we see much less SMAD2 and SMAD3 phosphorylation, meaning they have a lower activity here, meaning they have lower fibrosis. So now in this B cell now mice, now we give them IgG back. We restore IgG levels. What happened? With this IgG accumulation, again, in adipose tissue, we see huge upregulation of F480, which is the macrophage marker, meaning adipose tissue inflammation is increased. Also, we increase the SMAD2 and SMAD3 phosphorylation, indicating that we also activate the fibrotic pathways in adipose tissue by IgG. So long story short, the mechanism is figured out. We found it mediated through macrophage. At the beginning, we thought it directly target fibroblasts to induce fibrosis. Then it turned out we were wrong. So it requires macrophage. IgG can activate the macrophage. Macrophage then produce more TGF-beta that activate the fibroblast to make cause tissue fibrosis. Then the next question is, so what regulates IgG accumulation during aging? I showed you that it's unlikely it's B cells. We're going to touch this more carefully in details later. And now, so we look at another mechanism to regulate IgG. That is recycling. So IgG usually is uptaken by the cell, then it's in these small vesicles called endosomes. When endosome matures, its pH lower, it becomes more acidic. It's something very interesting happened now that at this acidic environment, IgG is handled from its receptor to another protein called FcRn. So then the FcRn bind the IgG at this lower pH, then transport IgG back to the plasma membrane, release at a neutral pH. In this way, IgG is recycled. Therefore, IgG can have this super long half-life. It's the longest in all antibodies. And people also know that the recycling rate is about 1.5-fold as the production by B cells. Therefore, IgG level is determined by its recycling receptor, FcRn. So we know macrophage is critical for IgG recycling. Therefore, we knock out, we just ablate specifically in macrophages of this FcGRT, which is the gene encoding FcRn. So after we abolish IgG recycling in macrophage, we shorten IgG half-life. That's as we expect. This model works. Good. Let's see what happens. So this macrophage knockout mice that they have no phenotype at a young age. They have a normal IgG. However, during aging, that they prevent IgG accumulation during aging. So in the aged MKO mice, they have a lower IgG, then they have a much lower activation of SMAD2 and SMAD3. That means they have much lower fibrogenic pathways. So look at their tissue fibrosis. They have a very healthy adipose tissue with much less fibrosis. So indicating that once we prevent IgG accumulation, we can prevent a fibrosis in aging. Then what happened to other parameters? So we know that during aging, that our metabolic rate decrease. So in this mice with the prevention of IgG accumulation, they can maintain higher level of oxygen consumption, meaning they can have a higher level of metabolic rate. They are protected from obesity. And also in aging, we develop insulin resistance that lead to type 2 diabetes. In this mice, at 15 months old, these mice are still very healthy. They are very insulin sensitive. They can clear glucose much more efficient from their blood. Also, once we keep the mice, let them naturally die, we notice that indeed they actually have a prolonged lifespan over 12%. So this 12% is pretty significant in mice. So to put this together, that preventing IgG accumulation extends both health span and lifespan. So this story being published about 1 year ago in Cell Metabolism. So we found that IgG accumulated since early aging, very early, equal to about 30 years old in humans, preferentially in adipose tissue. In there, it acted the macrophage, caused adipose tissue pathogenic remodeling, and then impair metabolism. Once we prevent IgG accumulation, we can increase health span and lifespan. We are so happy to see that about 2 months ago, that there's a study come out from a Cell that they use state-of-the-art technology, did a very comprehensive analysis across different species. They reproduced our finding that IgG is an aging factor that causes senescence. And they also found reproduce our finding that once we intervene IgG accumulation, we can improve aging. So we are so happy to see that our finding is reproduced. Now let's come to the second part. The first part I show you IgG in aging. Now what's its connection in obesity? As I mentioned to you before, that aging and obesity are so similar to each other, especially in adipose tissue, that they have the same hallmarks, including inflammation, fibrosis, insulin resistance, compromised adipogenesis, and senescence. They all lead to metabolic decline. So we ask, is IgG also a driving force for obesity? So in this case, we use McDonald's food to push mice to become obese. We call it a high-fat diet. In this case, that the IgG accumulate in adipose tissue again, and also there's no increase in IgA and IgM. This accumulation of IgG in aging and in obesity in adipose tissue is very specific to IgG, but not other antibodies. This accumulation in adipose tissue is also way higher than its increase in the plasma, meaning that adipose tissue is can suck all the IgG or accumulate IgG from the circulation. It's not just a leakage. The increased IgG include all the isotypes of IgG, IgG1 to 2A, 3. That's implying that it might be a polyclonal response rather than a monoclonal response. So what caused the IgG accumulation here? At the beginning, we thought it might be B cell because we know in age and in obesity, the antibody produced B cells can infiltrate in adipose tissue. They already cause the inflammation. So we use this B cell null mice, which has no IgG production. We give some purified IgG. Let's see what happened. Something very surprising to us. We noticed that this exogenous IgG specifically goes to adipose tissue. This the belly fat, subcutaneous fat, but not much to other tissues. So these antibodies are isolated, purified from another mouse, not is generated by the B cells, because these mice have no B cells, meaning that the IgG accumulated in adipose tissue in a B cell independent way. Also, we know that macrophage is critical for IgG recycling, as I mentioned to you before. So we may say, okay, this might be related to macrophage infiltration. Then we did compare the time course to see how fast IgG accumulate in adipose tissue in comparison to macrophage infiltration. We use the F480 as the marker. So we know that macrophage actually infiltrate in adipose tissue at a much later stage after obesity is established. However, IgG accumulated in adipose tissue in a much earlier time point, at even 2 weeks, when we put the mice on high-fat diet. So meaning that IgG has another mechanism to enrich in adipose tissue. So long story short, we did a series of conditioned mice. We showed that this depends on their recycling by different cells. So for function-wise, so once we prevent IgG accumulation, when we induce obesity, there is no IgG accumulation in adipose tissue. Then these mice were protected from diet-induced obesity. They also protected from insulin resistance that come along with obesity. So these mice are super metabolic healthy. They are free of obesity and insulin resistance. So what underlies this metabolic protection? So in adipose tissue, the very fundamental function is adipogenesis. That's adipocyte formation. We always need the adipocyte new adipocyte formation and turnover. So this determine adipose tissue remodeling, metabolic function, hypertrophy, and inflammation. So this pathway from pre-adipocyte to adipocyte is tightly regulated by different hormonal signal, especially insulin signaling. So we checked whether IgG has an effect on adipocyte formation. So we did this in vitro, isolate adipocyte progenitor cells, then differentiate them into adipocyte in the presence of IgG. So again, we were very surprised to see this finding that IgG itself can inhibit adipocyte formation. Here we use oil red O to stain lipid droplet in the cells. The beautiful red color here. So IgG treatment strongly reduce the lipid accumulation. Now you may see there may be some contamination in the IgG solution. So then we boil the IgG, we denature IgG, then leave all this, maybe some other small chemicals still remain intact in the solution. Let's see what happened. Then after boiling, that the denatured IgG lost their ability to inhibit adipocyte formation. And so for the pathway analysis, we found that IgG treatment indeed impaired insulin signaling during adipocyte formation, which is essential for adipogenesis. So insulin. So we are very familiar with insulin. There are thousands of people inject insulin every day. So once you inject the insulin, it can activate its receptor called insulin receptor to turn on all the downstream consequence. We did a computational modeling. We found that IgG, this purple molecule is IgG. This green one is the insulin receptor. Combined with the insulin receptor and at the same position of insulin binding. This is unbelievable to us. We didn't believe it. So similarly, if this is true, that meaning that insulin and IgG can compete with each other to bind with the insulin receptor. So we didn't believe the computational modeling, so we did an experiment to test it. Then we did the binding assay. We did find that insulin bind with its receptor at a high affinity, but in the presence of IgG, its binding is impaired, it's dropped several fold. That did tell us that insulin and IgG compete with each other to bind with the insulin receptor. We still didn't believe what we see here. We further tested it. So we used another approach that we put a small tail on insulin receptor that it can mark the protein interacting with it. So we call this called a turbo ID technology. With this, we saw that this labeled insulin receptor can mark IgG, meaning that they two did interact. So we did a further careful analysis. We identify a domain called on insulin receptor, which is called immunoglobulin-like domain 1. This a domain function is for interacting with the protein have the similar domain. Immunoglobulin is the protein have the very best candidate have this immunoglobulin domains, right? So after we delete this domain IGLD1, we call it IR.1, we abolish the interaction of IgG and the insulin receptor. This experiment convince us that IgG and the insulin receptor do interact and at the IGLD1 domain of IR here. From the textbook, we know that antibody need to bind with antigen to function, to activate its downstream receptor, Fc gamma receptors. So this is the classical activation. So we also say, okay, let's remove this antigen recognizing domain and see whether it can still function. So we use this constant domain of IgG. We treat adipocyte. Again, we inhibit adipogenesis through inhibiting insulin receptor. This tells us that IgG impede IR activation, not through its antigen binding, but through its Fc domain. This actually is consistent with our computational modeling, in which we found that it's through a smaller region on the Fc domain called CH3 to compete with insulin. Last, we want to see, okay, now we know that IgG is underlying both aging and obesity to cause the metabolic dysfunction and the inflammation. Then can we target IgG pathways to rectify or improve metabolic function? There are multiple ways to intervene IgG. There is one famous way or popular way is to deplete the B cells through antibody. Then the entire body cannot have enough antibody to protect us. So if we found through another way, we can target its recycling receptor, FcRn. In this case, we use antisense oligo. We treat the aged mice with this antisense oligo. We can decrease their body weight. We can improve their insulin sensitivity. We can also decrease their fibrotic genes in their adipose tissue and increase their genes involved in lipid oxidation, lipid utilization. We also checked their brown fat, in which this fat is very unique in mice that its function not for storing fat, it's rather for burning fat. This fat actually under a process called whitening, become white, lost their fat burning ability during aging. Well, by abolishing IgG accumulation, we can inhibit their whitening, make this fat much healthier. As a result, these mice have a higher oxygen consumption in their age. We also repeat this same experiment in obese mice. We can also improve the metabolic function, meaning that this is also a common therapeutic strategy to target both aging and obesity. So to put all this together, we propose that IgG is situated in the center of adipose tissue pathogenic remodeling that involve these all major players like adipocyte, fibroblast, macrophage, B cells, all connected by IgG. So it is an integrative pathogenic factor. From the big pictures, that IgG is accumulation is both happening in aging and obesity, can connect with all these common hallmarks involved in including impaired adipogenesis, inflammation, insulin resistance, fibrosis, and senescence. So all these functions are distinct from our classical view of the immune protection function of IgG. So therefore, we claim that this is the non-canonical function of IgG in aging and obesity. It also not functions through its antigen recognition, rather it's through its Fc fragment. So it's a function in a different through a different mechanism. At the end, I want to try to convey this message to you that we believe aging have a metabolic origin. So we know that our the lifespan is determined by many aspects. So many people are interested in identifying aging hallmarks in very late stage of life, then try to intervene these hallmarks to extend the lifespan. Indeed, the metabolic decline starts much earlier. So like in humans, after 30 years old, when IgG start to accumulate, that metabolic decline already start. This metabolic decline, the key to determine health span. And for this decline, we believe or we identify that IgG accumulating adipose tissue is a driving force. Therefore, so I propose to you so from a metabolic perspective, so we should start aging and intervene aging from a much earlier time point. So I want to thank all the people contributing to this study. So my life at Columbia and also at Peking University. Also my collaborators from Columbia and also from other institute, and our funding from NIH and also the China funding resources. I will thank you for your attention.
Thanks, Lee. That was great.
I look forward to your questions, Mike.
Yeah, so that was fantastic. So I have a whole bunch of questions, comments, suggestions, and all that. So just to put, okay, first the superlatives, right? I think that your data and the MA study that just came out too in November, this is the early days that I think the majority, even for the lay public, but even for the academic community, researchers, either are underrating the IgG story in health span and longevity, or just don't simply know. And I think your lab's work and the Moss study, their work, is going to be the centerpiece for maybe decades of the field having to catch up and further expand this story as a big player in aging and longevity. So with that in mind, just to put your data into further perspective. So when you think about blood the circulating milieu, right? So you've got red blood cells, which are the most abundant cell type. About 45% of the blood is red blood cells. Then you have the white blood cells, which are about 1%. And then you have the remaining 54%, which are the circulating proteins, metabolites, lipoproteins, etc. So to put the IgG data into perspective, IgG levels in blood are about 1,000 milligrams per deciliter. So this is one of the biggest trees in the blood forest. For people who may not know, and I'm sure you do, obviously, glucose, for example, in youth is 80 milligrams per deciliter. So we have IgG at 1,000, and 1,000 may be even an average or the median value, milligrams per deciliter. You have glucose at 80. Already, it's one of the biggest trees because it's 12 times higher than glucose. Okay, so now total cholesterol. Total cholesterol, let's say 200 milligrams per deciliter. IgG in just the average or median values without assuming aging or obesity, 1,000 versus 200 is a five-fold higher amount of IgG just than the normal state. So in terms of it being one of the biggest trees in the forest and not talked about in terms of glucose or lipoproteins, which are the dominant theme in health and health span, I think you're onto something fantastic that people are going to be studying for decades. And so anyway, that's what are your thoughts about IgG is one of the biggest trees in the forest?
So, Mike, I really appreciate your comments and your points. I fully agree with you. So this IgG amount, as you mentioned, is the biggest tree in the forest. It's so high, sky high. And it's actually there is an interesting story that at the beginning, when we did the mass spec, try to identify all the protein change in adipose cytokine aging, we didn't get it from the result. Then we went to talk to the people who run the mass spec for us. The people were so nice, so helpful. They told us that they filter the most abundant protein out at the background. I said, no, no, no. It's present there. If they change so much, it must be for some reason. So I guess that might be the reason why people didn't notice it in the last several decades. That there are many studies, elegant studies published in the top tier journals. They did a bunch of they analyzed plasma from different species, from humans, and it didn't present there. So I think they're too abundant there. So
Yeah, to your point, almost all of well, actually, all of the proteomic studies that I've seen in terms of aging and disease, like you said, they remove albumin and the globulins because there's such a dominant tree in terms of concentration in the forest that you don't see any of the smaller abundant proteins like cystatin C or TNF alpha, GDF15, etc. But yet, how are you throwing away the biggest trees in the forest? This is probably where the biggest part of the story is.
yes, yes. So that's therefore I will see that it's so interesting to us that aging is we all know aging, and then we also know IgG, very old topic, but they are all at very early age to study to put them together.
Definitely, yeah. All right. So then the other one of the thoughts is that so I think there may be a prequel to your story, right? So I think there may be a story underlying your story. And what I mean by that is I don't, sure, there probably is some overreaction by the immune system, by B cells, plasma cells, macrophages, et cetera, in producing IgG during aging. So you ablate that and you get improvements in metabolic health, reduction, or increases in median lifespan. So there may be some overreaction of the immune system that's going on, but what's the trigger for the increase in IgG at all, right? So when you consider that the gut can become more leaky, right? Whether it's microbial products or actually microbes themselves translocating into the blood, couldn't it be possible that IgG becomes elevated in response to some amount of microbial leak to neutralize some of those microbial pathogens that are now in the blood? But because there's that cycle doesn't ever really resolve during aging and only gets worse, IgG levels continue to increase. Now, another aspect of that story is that could it be possible that IgG is following some of these microbes into adipose tissue? There is some published data that, at least in the intestine, mesenteric fat can act as a sink for microbes to prevent them from going elsewhere systemically. So, have you thought about looking at either the blood microbial burden, whether it's whole genome sequencing to look at all of the microbes that could be there in young versus older mice? And then, even in adipose tissue, have you considered looking at to see maybe adipose tissue, old versus young, has a higher burden of microbial products or microbial DNA that may be a part of this, an earlier part of this story?
So, Mike, so you raised a crucial question that has been intriguing me in the past many years. Why does IgG increase in aging and in obesity? So let me put it in this way that so when we age, so we may get exposed to more and more pathogens like microbes. So this may enter our circulation, reach some tissue like adipose tissue. It's highly possible. And also the mesenteric fat, as you said, is like a filter from the nutrient and also hormones or blood from the gut. So it's easy to accumulate this is bacteria. So it's highly possible that this is a cause of IgG increase. There's other possibilities. So the increase of the pathogen our body keep accumulating with more antibodies to deal with more pathogen. So that's as a defense mechanism. So next time when our body see it, we have something ready in the body to defend us, but have to keep accumulating that increase. The second is that over time, that might be some change in the modifications on antibodies, on IgGs. All the IgG have this glycosylation that is essential for its protein stability, for its function. So there are also some beautiful studies showing that the glycosylation also change antibody function, makes them from immune protective to pro-inflammatory. So I speculate that over time in aging, the glycosylation of IgG also change something. That's another reason. There's a third one I speculate is the microenvironment in our tissue. So there's something changes there, like immune cells or some matrix proteins that can trap antibody there. So that's why IgG prefer to go to adipose tissue to stay there. So all of this, I have no definite answer for this. So but they are all the key questions. I look forward to more expert to work on it to figure this out.
So I have more to suggest along that story. And that's actually a great answer. You say you didn't really answer, but I think that's a great answer because the idea that the immunoglobulin function declines during aging and our B cells, plasma cells, et cetera, potentially increase IgG expression to compensate for that, right? If you have some less functional, you make more. Okay. So then you could say, what's the evolutionary advantage of having the IgG interact with the insulin receptor, leading to an increase in blood glucose, right? So an increase in blood glucose makes the osmolarity of plasma higher, which is osmotic stress. Osmotic stress is a well-known antimicrobial strategy. It causes the bacteria to basically burst. So this could be, and I know I'm still on the microbial burden story, but this would be my understanding of, okay, more IgG, less efficient at killing or coating microbes and helping to kill them off. And then there's the further compensation with interaction with the insulin receptor, higher glucose, osmotic stress, kill off the bacteria. Now, the other additional part that I have there is, I'm sure you're familiar, or maybe not, I don't know, but fat cells can produce antimicrobial peptides like cathelicidin. So I wonder if the IgG going to fat and being specifically, I agree that I mean, I think it's definitely, you mentioned that, maybe specifically localize the fat for certain reasons. But as another reason, I think it could be possible that just like the insulin receptor, maybe there's an evolutionary advantage where IgG accumulated fat tissue produces more cathelicidin, more antimicrobial peptides, because immune system function is down. So now we've got these other potential pathways to potentially reduce our microbial burden. But the consequence of that is higher glucose, worse cellular function, aging cells, more fat cells, more pro-inflammatory products, immunosenescence, inflammaging, right? But at least we're staying alive. So what are your thoughts on those additional?
So Mike, you are a real expert on this. I like your answer. You already answered the majority of the question. So agree with you that the evolution is a part of the explanation. So, like in the wild, the very few animals have this opportunity to live long to age, and also to have actual food to become obese. So our body or the animals evolved to try to have this immune protection. So, but over time, in modern society, the human just can live longer and more food. So this is actually, I feel it's like a trade-off of immune protection. So our body didn't haven't evolved a machinery to to handle too many antibodies. So all this selection pressure is all try to have more antibody for more immune protection. So that's in addition to your explanations. And also for this, I'm also trying to understand why IgG try to behave like a native inhibitor of insulin receptor, and from a physiological perspective. So put it in this way. So when we culture cells, we can add some drug or remove some drug. We can make it absolutely no insulin. That's easy. But in healthy humans, under physiological conditions, it's never happens without insulin. There's always insulin present in our bodies. Insulin signal is always so sensitive. Even a small amount can activate the consequence. But in our body, at a certain time, we need to shut off insulin signal. So if there is no shut off mechanism, so the insulin signal will be linear to decrease, but still there to function. So I speculate that IgG actually present in the circulation function as a buffering system to create a threshold for insulin. So when insulin is below this threshold, no, okay, boom, it's shut off. The body need to now to produce more glucose, to induce lipolysis, to so that's to have this a switch, a threshold for that. Also, you also actually bring this up. I like to extend a little bit. So there is a theory called lipid spillover. So when our fat, adipose tissue, cannot handle or store so much fat in our body, the fat is going to go to be stored in other tissues, like in the liver, called fatty liver, like in the muscle or other tissues. This is called lipid spillover. That's called a lot of issues here. So I would say that there is also an IgG spillover here. IgG first go to adipose tissue to store there, and then when adipose tissue cannot store that much, it start to accumulate in other tissue. In this way, actually, adipose tissue sacrifice itself, protect other organs. So I also am eager to see that other people work on this to check IgG function in other tissues, like in the muscle, in the brain. And also, I would also like to speculate that this is like a discussion. I do not have the clear answer for this because this is also very early time to discover IgG as an aging factor. We do not really know much about it. So all of this is a discussion and my speculation I'd like to share with you and the audience. So since IgG can accumulate at such a high level in adipose tissue, and also in adult humans, so adipose tissue can be the biggest tissue in our body. For you, that's not a problem, but for many other people, adipose tissue can be over 50% of their body composition sometimes. So imagine that so high IgG levels, so much fat. So fat maybe store substantial amount of antibody in our body. So therefore, adipose tissue can maybe function like a reservoir of antibodies. It regulates the whole body and IgG homeostasis.
I think that's definitely true, but I think that there's a part of the story that's missing that other studies have suggested, that fat can produce cathelicidin, which is antimicrobial. And I
Yeah.
wonder if, you get the IgG accumulation in fat. Now you get a big burst of cathelicidin. And in support of that, cathelicidin, which is antimicrobial, is higher in people with a higher BMI, obese BMI. I can send you that paper. So
Mhm.
that would support, but whether IgG accumulation in adipose tissue precedes that, I don't, nobody's studied that, right? So,
Yeah.
all right. So the next thing is, okay, we know that from your study, the calorie restriction, so IgG was lower in calorie restricted mice. And then in the Ma paper, parabiosis reduced IgG to relatively youthful levels. And I think too, they looked at exercise training. IgG
Mm.
was lower. So these are three, well, calorie restriction and exercise training are easily, most people can do that, whereas parabiosis may be for advanced stage. But do you know of any other therapies or therapeutics that could potentially help keep IgG low over time? What are your thoughts on that?
So, Mike, this is a million-dollar questions. Um, so in addition to this three ways you just brought up, that there is also in the clinic, the people are developing FcRn antibodies, try to treat autoimmune disease. That way to keep IgG level low. So that I would predict that this approach would be also be applicable for treating aging and obesity associated disease. That's my prediction. And but this treatment do have we have to be really cautious about it because our body need a certain level of antibodies. So this antibody, they try to develop the stronger, the most efficient ones. This can really decrease our body immune protection. So we have to be really careful with this treatment.
Yeah, so that gets into the age-related changes for IgG, right? So I know that there were a few human subjects in your study, and you did see that increase in, I think it was plasma, that IgG levels increased. I did a deep dive into all the studies that I could find for how IgG levels, like large epidemiological studies, just looking at how does IgG change during aging. And there are just very few studies, in the hundreds, not thousands to even more. Honestly, I don't know then, aside from these very small studies, how IgG levels change during aging. I presume it increases, but I think, I think it's possible. I'd like to hear your thoughts where clearly it's low in youth. It probably peaks at midlife. And then as the immune system declines towards the end of life, there may be some, inverse U where it actually starts to decline towards the end of life, and then increasing susceptibility to death. So do you think it's possible that IgG levels decline towards the very end, last decade or two of life, or do you think it's just a continuous increase?
And Mike, so this we have to distinguish the IgG in the circulation and in tissue. So I believe in tissue, it may happen as you just described. It may drop in very old subjects. But here I really need to clarify this. So all the IgG measurement is mainly for circulating antibodies, not in the tissue, because tissue is not easy to measure. But to us, we feel that the IgG in the tissue counts, but not in the circulation.
Hmm.
So IgG accumulated in tissue is in a much more sensitive way and much earlier. So we should look at tissue IgG instead of circulating IgG here. I would say that is a better indicator, more accurate indicator.
Could you have higher levels of, I mean, probably you want to measure in adipose tissue, right? Since that's the biggest reservoir for IgG.
Mhm, mhm.
So wouldn't it be proportional where high blood IgG would be proportional to high body fat? Or is there a situation where you could have low body fat, but low body fat, low visceral fat, but high blood IgG? Wouldn't they be proportional? What's in the blood should be proportional to what's in fat?
I thought about this. So like in different tissues, they have the same level of IgG in the circulation, but they have a very different levels of tissue IgG levels.
Mm.
And also, like in the very later stage of life, actually, people body fat loose, they may have higher IgG in their fat. And some people that they may be obese, they have more fat. These people can be very healthy. These people are highly speculated that they have a less IgG in their adipose tissue. And also you just mentioned that exercise, calorie restriction, and periodic can also change IgG levels. Meaning IgG level in adipose tissue can change quite efficiently. It's not I would say they're constant. So there should be some way to manipulate IgG, and it's not necessary in this proportional way to fat.
Okay, got you there. So in terms of the high-fat diet, so you saw a big spike in IgG, high-fat diet. So I'm guessing the high-fat diet was the purified, just the standard purified, cellulose as the fermentable fiber, right? So when you think about it within that context, a high-fat diet, even though it's obesogenic, it's in part obesogenic because it lacks soluble fiber. It lacks fermentable fiber. So I wonder if you take the purified chow, even though it's not chow. Chow is, corn, wheat, and oat based. But so if you take the purified diet and add soluble fiber back in, I wonder if you see a reduction in IgG in the high fat diet fed mice. What are your thoughts?
This is an interesting idea. It's highly possible. I would say that the higher fiber would change or benefit metabolism and the immune function. That it may lower IgG, but without seeing the data, I'm not definitely have that definite answer.
And then
It's all
when you,
just need more research. What is
yes,
simple?
100%. But it would have to be soluble fiber, not more cellulose. More cellulose won't do anything because it's not fermented by gut bacteria. So if you put them on the high fat obesogenic diet and you give them soluble fiber, pick your soluble fiber, inulin, pectin, I can go down the list, resistant starch. Would that make an impact on, I mean, for sure, it would probably improve metabolic health. That's well known in soluble fiber supplementation in high fat diet studies. But would it bring down IgG? As far as I know, nobody's done that study.
Yeah.
Yeah. So
Really need people include IgG in their measurements for
yeah,
this.
like I said, you guys are on the cutting edge. Most labs probably won't do that for a long time. So, all right. So IgG is a broad class, right? You've got IgG against Candida, against CMV. I mean, we can go down the list, right? So, is that pattern different in aged mice than it is in humans? And are there, or which ones are specific? Are they all globally increasing, or is it specific IgGs to specific microbes that are increasing?
And for that, so we do not know. We haven't compared this IgG antigen specificity yet. Um, so I think there must be difference in humans and in mice. But bear in mind that we find that IgG function through this Fc fragment, not through this antigen recognition or through the FAB domain. So we argue that it's not through IgG antigen specificity, but it's simply the amount of IgG.
So the global, probably the most important, not necessarily the specific components of the IgG class, but couldn't it be possible that there are specific, like maybe it's IgG for Candida is way up, but then the others there are no change. And if you know that, then you go after potentially where the source of where that can, if it is with Candida in mice, I don't know, I'm just speculating, it could be anything. But couldn't you then target, all right, we know that IgG spiked against Candida in mice. If we're going to give some therapy to get rid of the candida, does the systemic IgG come down in metabolic health, right? So it goes back to that prequel idea.
It's possible. It's highly possible. But I have to keep reminding you and the audience that so in the past many years, people believe or look at IgG that it must function through specific antigen. But I think that's the way people maybe also missed this point that IgG not necessarily function in this canonical way. So
So,
I in my very end slide I put there that we claim that this is the non-canonical function of IgG.
well,
So
so I think what you're saying is it's not so much important to look at the individual IgG against CMV, Candida, whatever. It's the global, it's the total amount of the IgG from all of it that is the most important.
yeah, imagine that so we did that experiment. So we isolate IgG from another young healthy mice. They don't have this specific antigen to like CMV or to whatever, but just increased amount in another healthy mice. It caused all the issues. So very simple.
Wait,
It's
you took IgG from healthy mice and transplanted it into,
another healthy mice.
wait, what if you transplanted those healthy IgG into the old mice? Have you done that experiment?
So if we just increase the boost the amount, the level of IgG, we already have these issues of inflammation and the metabolic dysfunction. So therefore we didn't try to transfer young antibody to an old and aged mice. So I would say that we just make it worse if
Yeah.
we further increase IgG levels.
Yeah. Okay. So current studies, are there current studies that you guys are doing and then future studies that you'd like to share ahead of time?
Yes, a little bit. So we know that from this very unexpected, surprising finding to us, we are expanding to establish this non-canonical function of IgG in aging and obesity associated comorbidities. We just would to like a pre that to just to tell you that so we have some very striking findings that IgG also cause other issues. It's coming up. Please stay
Nice.
tuned.
Nice. Well, I'm looking forward to it, and hopefully we can have you back in on the channel at some point to update us after the paper is published. So once again, thanks, Lee, and hopefully we'll see you again soon.
Thank you so much, Mike. This is you are really doing a great job and then have this very unique platform to share to connect the scientific research community with the public. That's critical. I
So
really like the idea like